Hello, in this video we will discuss about the blood-brain barrier. This is structure and function we will discuss in this video. So what is the blood-brain barrier? We will learn all these things in this video. So remember, the blood-brain barrier is triple B is a semi-permeable and extremely selective system or most vertebrates that separates blood from the brain's extracellular fluid. So the extracellular fluid will be separated through blood-brain barrier. While on the other hand, the cerebrospinal fluid barrier is a separate things we will discuss in the next video. So this is a blood-brain barrier. This is the blood circulation to the brain, and this is the brain, and this is the blood vessel. This blood vessel is basically surrounded the brain and in the form of capillary. So this is the capillary I am drawing, which that is contain astrocytes here, and this is the brain, and inside is the blood. So the blood, the concentration regulation will maintain due to the blood-brain barrier, and the orange color is showing that the here is the tight junction, and orange color shows that the basement membrane, and this is the astrocyte, while this endothelial cell of the capillary, which that's surrounded by the basement membrane as well as astrocyte. So this is the concentration regulation is very important in the brain. So the human brain has 644 kilometers of blood vessels provide oxygen, energy, nutrients, metabolites, and other contents to brain cells, while also removing carbon dioxide as well as metabolic waste. So the brain total body mass is 2%, but the glucose and oxygen consumption is 20% required. It is exchanged through blood-brain barrier. So remember, important thing is that the brain consuming more energy throughout the body for normal function, normal physiology, while the several part of the brain which that will not require the blood brain barrier. For example, when the pituitary gland, which that is part of the brain, release the hormone directly into the blood vessel, into the blood circulatory system, and this is the cerebrospinal fluid will support, while the circumventricular organs are present middle region of the brain, and which that require no blood brain barrier. So the cerebrospinal fluid is very important for it. So let's begin to understand the anatomy in the transverse section of blood brain barrier. So this is we can get the more knowledge from this. So this is the inside is the endothelial cell, which that's contain capillary, and this blood capillary middle side, while this is the orange color is showing that the neurons, and this is basically the brain, while here is the blood capillary, which that's contain red blood cell and white blood cell. So this is the astrocyte. Remember, this is also supportive, and in this way here is the orange color layer is showing that is the basement membrane. So this basement membrane is very important, as well as on the other hand the tight junction between the endothelial cell are also very important for the prevention of the passive transport of the nutrients. So this is the microglial cell. This microglial cell, and here is the tight junction. Microglial cell, which that is used for the immunity, protection, and phagocytosis of the pathogen antigen. So this is the immune response due to the microglial cell as a macrophage like structure are present in the brain for prevention of the disease and invagination of the pathogen protection. So this is the parasite also present. This parasite is the orange color show, and on the other hand, here is the astrocyte is used for the migration of new developing neuron and buffer the potassium and neurotransmitter buffer. So this is the buffer is very important for the normal function of the neurons. So let's begin to understand. Here is the tight junction. What is the tight junction and between the tight junction? So this is the paracellular space, and this is the basolateral plasma membrane, while this is the apical plasma membrane outside, and this is the thin tight junction 50 to 10 times closer than others endothelial cell. So the squamous and endothelial cell structure. So here the zoos is the zonula occludens and heterotrimeric protein found on the neighbor cells. So let's begin to understand what is the type of protein are present. So this is the basically is the zonula occludin. Basically zonula occludin 1 to 3 types. We will not understand with detail, but you can understand. And here is the zo1 and zo2 protein will bind with the actin for the complete contraction, and to basically actin will bind. And in this way here you can see this is the another protein, the claudin and the occludin. Basically the claudin and occludin, which basically is used for the also the support for the tight junction. So in this way the claudin 5, 3 and 12, which that elevated the transendothelial electrical resistance, and claudin 5 is required for the tight junction development and the blood brain barrier function. So what is the transendothelial electrical resistance? So on the other hand, here is the junction adhesion molecule ABC. This is used for the junction, the tight junction, the junction adhesion molecule. So in this way, the junction adhesion molecule ABC is used for the transendothelial electrical resistance TEER. And in this way, when the junction adhesion molecule ABC, which that is junction adhesion molecules and specific transport mechanism for limiting the polar substance, passive transport inhibition, while active transport can be promote. So in this way, I hope you make sense. This is the tight junction due to the junction adhesion molecule, and claudin and occludin, which basically is the transendothelial electrical resistance and is required for the tight junction development and blood-brain barrier function through claudin. So here is the pericytes, which that is a function of pericyte we will discuss. So the brain capillary pericytes are located in the center between the endothelial cell. So this is the phagocytic activity linked to clear the antigen. So that is very important, the pericyte. Pericyte is very important for the phagocytic activity linked to clear the antigen, also like a microglial cell. But in this case here you can see this is the tight junction are present between the capillary and the basement membrane are also present, which that contain a many type of content. And on the other hand, here is the basement membrane, endothelial cell and pericyte, and this is the extracellular matrix and intracellular matrix within the endothelial cell. So this is the tight junction between the endothelial cell and the pericyte is present. So this is the basically the collagen, nitrogen, laminin and S proteoglycan and G protein, which that is the composition of the basement membrane. So that is why this is contain a complex substances. So let's begin to understand the endothelial cell, which that contain alpha beta integrin receptor in the outside with link with the basement membrane, because basement membrane contain very collagen, nitrogen, laminin and S proteoglycan and G protein. So this is the blood, and inside here is the interaction with alpha and beta integrin receptor to the basement membrane with the receptor this. So interaction will through this alpha and beta integrin receptor. So let's begin to understand the blood-brain barrier materials transmission across blood-brain barrier. So this is the blood and inside is the brain, and in this way here is a water soluble molecule can diffuse directly where the junction is present, and the paracellular diffusion it is known as, while the lipid soluble can be diffused with the cell to cell, and the transcellular diffusion pathway is this. And in this way here you can see the amino acid, glucose and nutrient can be diffused through a transporter, and this is known as the transporter protein, which that is used for the transmission of the content nutrients. So here is the receptor mediated transcytosis, which the receptor will bind with the drug insulin, will endocytose. After the endocytosis will move or transmit into the brain for the normal function, for the binding of the insulin with the insulin receptor. And here the absorptive transcytosis, which that is the negative and positive charge. So the negative charge in the plasma membrane will attract the nanoparticle or macromolecule, will engulf and move into the brain, is known as the adsorptive, not absorptive but adsorptive transcytosis. So the function of the blood-brain barrier is the sodium chloride and water, for example, regulation, prevent to swelling. If the blood-brain barrier break, the water and sodium move into the brain and swell the brain, lead to create high pressure in skull and cause Alzheimer, epilepsy causes, and on the other hand, Alzheimer disease, and on the other hand the trauma and shock or stroke, and which that will lead to cause the death. So that is important things, which that if the blood brain barrier will break, so the microbes and pathogen can invaginate, will cause the infection into the brain, and that brain causes of the infection will lead to cause the other problems. So in this way I hope you make sense about these things. This video about the blood brain barrier, the structure and function. So if you like this video, please make sure to like, subscribe and share to your friends, and give the support via sharing this video, and give the suggestion for upcoming video. So thanks for watching. Bye.